Assalamu alaikum. This is Waliullah, student of MLT 8th semester. Today, my presentation topic is about enzyme-linked immunosorbent assay, that is ELISA. Outline of the presentation: introduction, basic principle of ELISA, requirement of ELISA test, procedure of ELISA, types of ELISA, interpretation of ELISA. Introduction to ELISA. ELISA test stands for enzyme-linked immunosorbent assay. It is a type of serological test and immunoassay technique. In the ELISA test, an enzyme links to the antibodies, particularly to detect the presence of protein-like antigen. The ELISA method was evolved from the radioimmunoassay technique in the 1960s. By the increasing concern over environmental pollution, the ELISA technique has been introduced instead of radioisotopes. Therefore, ELISA is a widely used method for the detection or diagnosis of bacterial and viral infections. Principle of ELISA. ELISA is the immunoassay technique which comes under the category of labeled immunoassay. Immunoassay depend upon the interactions between an antigen and antibody, which forms the antigen-antibody complex called immunocomplex. ELISA, being an immunoassay method, is very sensitive and specific in nature, whereby the specific antigen or antibodies bind to their homologous antibodies and antigen respectively. The enzyme converts a colorless substrate chromosomes to a colored product, indicating the presence of antigen and antibody binding. Requirement for ELISA test. The following are required while performing ELISA. Purified antigen when we detect or quantify antibody in a sample. Purified antibody when we detect or quantify antigen in a sample. Standard solution. Sample to be tested. Microtiter plate. It is made up of polystyrene which contain 96 well. Wash fluids. Enzyme labeled antibody and enzyme substrate. ELISA reader, that is spectrophotometer, for quantitative measurement. Procedure of ELISA. In ELISA assay, the antigen is immobilized to a polystyrene plate, which is a solid surface, and is attracted or has an affinity toward bacteria, other antibodies, and hormones. This is done either directly or by the use of a captured antibody itself, immobilized on the surface. The antigen is then complexed to a detection antibody, conjugated with a molecule for detection such as an enzyme or fluorophore. Finally, the substrate is added. The substrate is converted by the enzyme to form a colored product, which can be measured by spectrophotometry. Types of ELISA. There are 4 types of ELISA on the basis of binding structure between the antibody and antigen: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. First, direct ELISA. Antigen is immobilized onto the well of a 96 well polystyrene plate via passive adsorption. An enzyme labeled primary antibody specific for the target antigen is added to the well and directly bind to the antigen. A respective enzyme substrate is added, which upon reaction with the enzyme produced a visible calorimetric output that can be measured by a spectrophotometer or absorbance microplate reader. This is the diagram in which ELISA procedure is shown. This is the polystyrene plate. First antigen is immobilized on the polystyrene plate. Then primary antibody conjugated with enzyme is added which bind with antigen. Then substrate is added which convert by the enzyme into a color product. And this color product then detected by spectrophotometer which gave the result of ELISA. Indirect ELISA. In indirect ELISA, the antigen is bound to the bottom of the microplate film. Then an antibody specific to the antigen is added. A secondary antibody conjugated to an enzyme or other detection molecule is then bound to the first antibody. This is the diagram in which indirect ELISA procedure is shown. This is a polystyrene plate on which antigen is immobilized. Then primary antibody is added which is not conjugated with enzyme. This primary antibody is bound with the antigen. Then secondary antibody is added which is conjugated with enzyme. This secondary antibody is bound with the primary antibody. Then colorless substrate is added which is converted by this enzyme to a colored product. This colored product is measured by a spectrophotometer which interpret the ELISA test. Procedure of ELISA. In the first step, coat the microtiter plate well with antigen. The second step, block all unbound sites to prevent false positive result. The third step, add sample containing antibody to the well and incubate the plate at 37 degrees centigrade. The fourth step, wash the plate so that unbound antibody is removed. In the fifth step, add secondary antibody conjugated to an enzyme. In the sixth step, wash the plate so that unbound enzyme linked antibodies are removed. The seventh step, add substrate which is converted by the enzyme to produce a colored product. And the last, reaction of a substrate with the enzyme to produce a colored product. Sandwich ELISA. For the sandwich ELISA, two antibodies specific to different epitope on the target antigen are used. The captured antibody is bound to the bottom of the microplate well and bind one epitope of the antigen. The detection antibody binds to the antigen at a different epitope and is conjugated to an enzyme that enables detection. This is the sandwich ELISA procedure. This is the polystyrene plate on which primary antibody is immobilized. Then antigen is added which bind with the primary antibody. Then secondary antibody is conjugated with enzyme is added which make a bond with primary with the antigen. Then colorless substrate is added which is converted by this enzyme to a colored product, and this colored product is measured by spectrophotometer. Procedure of sandwich ELISA. Prepare a surface to which a known quantity of antibody is bound. The second step, add the antigen containing sample to the plate and incubate the plate at 37 degrees centigrade. The third step, wash the plate so that unbound antigen is removed. In the fourth step, add the enzyme-linked antibodies which are also specific to the antigen and then incubate at 37 degrees centigrade. Procedure of sandwich ELISA. Wash the plate so that unbound enzyme-linked antibodies are removed. In the fifth step, add substrate which is converted by the enzyme to produce a colored product. In the seventh step, reaction of a substrate with enzyme to produce a colored product. Competitive ELISA. This test is used to measure the concentration of antigen in a sample. In this test, antibody is first incubated in a solution with a sample containing antigen. The antigen-antibody mixture is then added to the microtiter well which is coated with antigen. Competitive ELISA. The more the antigen present in the sample, the less free antibody will be available to bind to the coated well. After the well is washed, enzyme conjugated secondary antibody specific for isotope of the primary antibody is added to determine the amount of primary antibody bound to the well. The higher the concentration of an antigen in the sample, the lower the absorbance. Procedure of competitive ELISA. Antibody is incubated with sample containing antigen. In the second step, antigen-antibody complex are added to the microtiter well which are pre-coated with the antigen. The third step, wash the plate to remove unbound antibody. In the fourth step, enzyme-linked secondary antibody which is specific to the primary antibody is added. The fifth step, wash the plate so that unbound enzyme-linked antibodies are removed. In the sixth step, add substrate which is converted by the enzyme into a fluorescent signal. Interpretation of ELISA. The result could be interpreted in the following two ways. Positive ELISA. If the enzyme-linked antibodies bind to the specific antigen, then this will give a positive result for the ELISA. We can neither see the interaction of antigen and antibodies nor the presence of antigen or antibody. Therefore, the colorless substrate is added to the wells where it reacts with the enzyme that are linked to the antigen-antibody complex and later release some signals in the form of color change. Hence, a positive result is indicated by a color change, and higher is the concentration of antigen, higher is the signal produced by the substrate. Negative ELISA. If the enzyme-linked antibodies do not bind to the antigen or there is no such interaction of antigen and antibody, then it gives the negative result for ELISA. Therefore, in negative ELISA, we can neither see any color change by the addition of substrate nor any bonding between the antigen and antibodies. Therefore, we can say an ELISA is basically a colorimetric assay which is detected by any color change in the well to conduct the presence or absence of antigen finally. Thank you so much.